All right, good morning, everyone. Is this where we left off? Okay. So we talked about coatings on the outside of the cell wall. So some bacteria, in addition to the cell wall, have other protective measures. They can have an S layer, which is basically a protein shield, or they can have something like a capsule, which is a sugar coating around the outside of the bacteria, and it protects them from drying out or from being phagocytosed by white blood cells. So Streptococcus pneumoniae, which causes pneumonia, is what's shown here. So the bacteria is just a little thing in the center, and the white area around it is the actual capsule. All right, so whether it's an S layer, or whether it's a slime layer or a capsule, we refer to this as the glycocalyx. And one of the functions of the glycocalyx is to allow cells, bacterial cells, to attach to a surface. This is medically important. Biofilms will form on things like catheters, medical equipment, and they can cause huge problems. They can cause nosocomial infections, which is a hospital-acquired infection. So you go to the hospital to get better, and instead the catheter that they stick into your vein ends up getting a bacterial contamination, and you end up having a more severe infection than what you even came in with. So this is important to anyone who's going into the healthcare field, is to understand what biofilms are and their role. So one of the biofilms you're probably most familiar with is just the plaque on your teeth. So you get that filmy layer on your teeth. Catheters, as I said, so the catheter is sterile when you put it in, but bacteria will start to grow on it over time. And what happens is you'll have a surface, a couple of bacteria come along and attach to that surface, and the green is the glycocalyx. That sugar that they're producing, I guess you can kind of think of it as syrup for your pancakes. It's a very sticky substance that allows the bacteria to be able to stick to each other as well as to the surface. And then over time, this just builds and gets thicker and thicker. Other bacteria may also come into this and become part of this biofilm. So why this is so important is the bacteria living in this biofilm are harder to kill than just free-floating bacteria. So the glycocalyx and the thickness of this prevents antibiotics from being able to get into and penetrate into this. And so even if you treat someone with antibiotics, the bacteria in here may not die because they're protected by all of this sugar molecules that are surrounding them. All right. You don't even need your web clickers for this one. So true or false, bacteria fimbriae are used for conjugation. Who thinks that's true? No? False? Don't know? A couple people raised their hand. All right. Fimbriae are not used for conjugation. Remember, conjugation is the sex pilus. Conjugation is the fancy term for bacterial sex. All right, we've talked about a lot of things on the outside of bacteria, the cell wall, the flagella. Now we're going to talk about some of the things on the inside. So the cell envelope is composed of the cell wall, but also the cytoplasmic membrane, as well as the outer membrane. So all bacteria have a cytoplasmic membrane. In fact, all cells have a cytoplasmic membrane. We also have the bacterial DNA or chromosomes. We have ribosomes for making protein, and we have the cytoplasm. So the cell wall, it helps to determine the shape of the bacteria. It provides structural support, keeps the cell from collapsing or popping if there's if the water pressure on it is too great. So osmotic pressure, hopefully you remember this from your chemistry class, if you have a lot of salt molecules inside the bacteria and a lot of water outside, the water actually wants to go inside the cell, and that causes the cell to swell, and it can pop if it doesn't have a cell wall to protect it. So the cell wall in bacteria is composed of peptidoglycan. Virtually all bacteria have peptidoglycan in their cell wall. And as the name suggests, peptide means protein and glycan means sugar. So it's a combination of protein and sugar. All right, so why would a cell wall be important for a bacteria but not for an animal cell? Why don't our cells have cell walls? Yeah.
The membrane around an animal cell is semi-permeable, so only like certain materials can even get in.
Bacterial cell membranes are semi-permeable too. Yeah. What happens if you don't like your conditions?
Oh, we can change them.
You can change them. If you're in a pool of water and you see your fingers get all shrivelly, like when you sit in the pool too long, you can just get out. The bacteria doesn't necessarily have that choice. So it needs a cell wall to protect it from environmental conditions that it has no control over. All right, the structure of the peptidoglycan. So this is important not just for understanding how bacteria work, but many antibiotics target the cell wall of bacteria. So the cell wall is sugar molecules that form chains and then amino acids or peptides that cross-link them. And so the result is you basically make like a fishing net structure that surrounds the cell. Many of the antibiotics that we use, penicillin, ampicillin, anything that ends in "-cillin", targets the production of these protein cross-linkers. So what happens is when you treat a bacteria with penicillin, these peptide cross-linkers go away, and instead of having a net, you just have a whole bunch of strands, and that doesn't work very well. Those strands just then fall apart, and the cell will pop or explode. There are two main types of cell walls in bacteria. We call them gram-positive and gram-negative. And when you use the gram stain in lab, you're trying to figure out whether the bacteria has one of these cell walls or the other. So gram-positive, the purple layer here is the peptidoglycan. So they have a very thick layer of peptidoglycan. They also have a few proteins or other molecules. Lipoteichoic acid helps to attach the peptidoglycan to the cell membrane. Gram-negative bacteria, on the other hand, the kind of darker pink layer here is the peptidoglycan in gram-negative bacteria. And notice that it's much thinner. It's not as thick as the gram-positive. And the other thing about gram-negatives is they have two cell membranes. They have the actual cell membrane and then they have an outer cell membrane. So you need to be able to distinguish between the two types of cells, gram-positive and gram-negative. And I could just show you a picture of them as an exam question and ask you to tell me which one it is. So you need to know what it looks like and what the differences are. Or I could give you a multiple choice question and say, which cell has an outer membrane and an inner membrane. So you need to know that that's the gram-negative. All right, we call it the Gram stain because in 1884, Hans Christian Gram discovered the stains that allowed us to do the Gram stain. All right, so the way this works is you add crystal violet, which is a purple dye. It turns all the cells purple. Then you add iodine. And then you wash all of the cells with alcohol. If the cells are gram-negative, if they've got just this thin layer of peptidoglycan, then when you add the alcohol, the purple stain leaves. So only the gram-positive cells with this thick peptidoglycan layer are going to stay purple. Then you add another stain called safranin, and the gram-negative cells then turn pink, and your gram-positive cells are purple. This is actually one of the most common initial diagnostic techniques used in medicine. So if a doctor takes a throat swab and sends it down to the lab, they'll just quick do a gram stain. Is it gram-positive or gram-negative? And the reason they do that is the antibiotics you use to target these two bacteria can be completely different. So antibiotics that are based on cell wall, the peptidoglycan, work usually very well for gram-positive, but they don't usually work very well for gram-negative. And so the doctor needs to know that almost instantly before he can prescribe an antibiotic. All right. So the thickness of the cell wall is about 20 to 80 nanometers thick, which on a microscopic scale is actually pretty thick. The lipoteichoic acids are involved in cell wall maintenance as well as anchoring the cell wall to the cell membrane. In the gram-negative cell wall, the peptidoglycan is only 1 to 3 nanometers thick. So it's like 10 times thinner in a gram-negative bacteria. There's pros and cons to this. One of the benefits is it makes the bacteria a little more flexible, so it can move a little bit more. All right, also in the gram-negative outer membrane, we have lipopolysaccharide. So on this outer membrane, the pink part, the outermost portion of the membrane, has lipopolysaccharide. So lipopolysaccharide, it's a lipid and it's a sugar molecule combined together. It's medically important though, because your immune system senses lipopolysaccharide. And if you get too much lipopolysaccharide in your blood, you end up going into septic shock. So if you've ever heard of someone going into septic shock, it's because they had an infection with a gram-negative bacteria, and there was so much of it that the immune system responded to this lipopolysaccharide, and your immune system actually just tears your whole body apart. All right, so we refer to lipopolysaccharide as an endotoxin. You could just take pure lipopolysaccharide and inject it into someone and kill them from septic shock, even if there wasn't a bacteria growing there. We also have lipoproteins that are in the outer membrane and porins. So we need to let molecules get through this outer membrane to the cell membrane, things like sugar molecules for energy, sensing your environment. Those things need to be able to get through that outer membrane. And so the porin proteins allow for things to pass through. They only allow small things though. So you can't shove like a whole protein through this. You actually have to shove pieces, peptides of a protein. One of the other things the outer membrane does is it blocks harmful chemicals from getting into the cell. So some antibiotics are too big to pass through these pores, and so they can't work on gram-negative bacteria. All right, then we have some bacteria who have kind of unusual cell walls. Mycobacterium, so this is tuberculosis. The cell wall of tuberculosis bacteria has what's called mycolic acid, and it's actually a wax. So it's basically waterproof, everything proof, and so people who have tuberculosis have to be treated with very specific antibiotics. Otherwise, they won't even pass through that waxy layer. So that protects the bacteria, so that's good. But it also means that things the bacteria wants don't pass through the cell wall very well either. So in order for a sugar molecule or something to pass through the cell wall, it takes a lot longer. And so mycobacteria actually grows really slow. So it's a two-edged sword. It helps keep the bacteria safe, but it means the bacteria gets nutrients much slower and it grows slower. So if you want to look for Mycobacteria tuberculosis, you use the acid-fast stain. So you use a different staining technique that can detect the presence of this wax or the mycolic acid. All right, Mycoplasma. This is a very small bacteria, and it actually doesn't have a cell wall. Oh, yeah.
Why do they call it mycolic acid if it's a wax?
It actually, so it has a very long hydrophobic portion of it, but it also has an acid on the end. So, all right, Mycoplasma. So these bacteria don't have a cell wall. That's kind of unique. They do have sterols, so molecules that are similar to cholesterol, and that helps to protect them from the environment. They tend to be about 1/10 the size of a normal bacteria, so they're only slightly larger than a virus particle. This right here is the Mycoplasma bacteria, and you can see it kind of looks like it has a funny little sucker on it, but that's just a specific shape at the end of the bacteria. They can be donut shaped, they can be long and filamentous, or they can be round and coccus. So because they don't have a cell wall, their shape can change because there's no structure there keeping them from moving. So Mycoplasma pneumoniae can cause pneumonia. That is a medically relevant type of bacteria. Well, you can't treat Mycoplasma with an antibiotic that destroys the cell wall because it doesn't have one. So if you treat someone with this disease with penicillin, you might as well treat them with water. It would be just as effective. So you need to understand the organisms that are causing disease in order to even know how to treat them. All right, let's do this clicker question quick. All right, what part of the outer membrane of gram-negative bacteria contributes to disease? All right, D is the correct answer. So lipopolysaccharide is in the outer membrane, and your immune system detects that. So it's actually your immune response that ends up hurting you if you have septic shock. The bacteria is, kind of bad on its own, but your immune system's response to the bacteria causes all the blood vessels in your body to simultaneously open, and all the fluid leaks out of your blood pressure drops, and you go into shock. So it's not the bacteria so much that's killing you as your immune system's response to the bacteria that kills you. All right, so let's go deeper inside the cell to the cytoplasmic membrane. So it's a lipid bilayer. 30 to 40% of it is phospholipids. 60 to 70% is protein. So there's actually a lot of proteins. So the blue shaped things here indicate proteins. So a lot of your cell membrane is actually proteins. So why are these proteins there? Well, some of them may be detecting signals. So in order for the bacteria to know where it wants to swim, it has to have proteins there that can sense the presence of sugar molecules or chlorine bleach or something that it wants to go to or away from. We also have proteins that allow things to get in or out of the cell. So we have transport proteins that will transport sugar molecules across the cell membrane. The cell membrane of all cells is selectively permeable. So some things pass through the cell membrane fairly well, and other things don't pass through basically at all. So actually water moves through the cell membrane fairly well. We think of the center of the cell membrane as being hydrophobic, and it is, but actually water passes through the membrane reasonably well. Glycerol, it's okay, but if you start looking at ions like chloride or potassium or sodium, those don't get through the cell membrane very well at all. And so if we want to pump those in the cell or pump them out of the cell, we need one of these proteins in order to do that, because the ion isn't just going to pass through the cell membrane normally. All right, so the cell membrane regulates transport, so we can control nutrients or waste coming in or out of the cell. It actually is where the electron transport chain is in bacteria. So this is where the energy is made. The cell membrane is the source of the energy for a bacteria. All right, there's three main types of transport systems. We have a simple transport system. We have group translocation and ABC system. All right, simple transport, we have a molecule. We want to get it inside the cell, and we'll use a hydrogen ion to get it inside the cell. So are there more hydrogen ions inside the cell or outside the cell? What was that? Outside. So the electron transport chain in a bacteria is in the cell membrane, and it actually pumps hydrogen ions outside the cell. So there's a lot of hydrogen ions outside the cell, and they want to come back in. So you can use the energy of that hydrogen ion coming back into the cell to transport something else inside the cell. You can have group translocation, which means that we actually modify whatever we transported inside the cell. Usually we add a phosphate group to it. That's what the P stands for, is a phosphate group. Or we have the ABC system, and it's not like ABC 1, 2, 3. ABC is an acronym. It stands for, ATP binding cassette. All right, there are very specific things that microorganisms want. They like iron. Iron is important in the electron transport chain. It's important for a lot of enzymes to function properly. Where do you get iron from if you're a bacteria?
Surroundings, soils, and...
Okay, most iron in the soil, though, isn't bioavailable. Like iron in a rock is useless to a bacteria. So really you need like iron ions. You need things that are dissolved in water. And so in order to get those iron ions, bacteria have molecules called siderophores, and the green in this molecule is where iron is grabbed by the siderophore, and then it is transported inside the cell. So free-floating iron is actually pretty rare. And so bacteria have evolved ways to be able to trap iron and get it inside the cell as efficiently as possible. All right, other things that are in cell membranes. We have secretion systems. So especially pathogens need to be able to secrete toxins and things. So the type 3 secretion system works just like a hypodermic needle. It's basically a syringe. You have a toxin or something inside the bacterial cell, and it will inject it through the syringe into your cells. So it's a toxin delivery mechanism in most instances. All right, the lipids in the cell membrane. These are phospholipids. So remember I said you needed to know the functional groups. Which functional group is this? A double bonded oxygen with a single bonded oxygen.
Ester.
A what?
Ester.
Okay, so this is an ester linkage. So this used to be a carboxylic acid or a carboxyl group, and now that it's bound here, it is an ester. The reason you need to know that is this is what is found in bacteria and animal cells, but in archaea, another type of microorganism, it's an ether, not an ester. And so you can tell the difference between a bacteria and an archaea just by looking right there in their cell membrane. So I could show you a picture of this. So this is what an ether looks like. Notice there's no double bonded oxygen. So this is an ester from a bacteria or a eukaryotic cell, and this is an ether from an archaea. So if I just showed you this picture, I would expect you to be able to tell me whether it came from a bacteria or an archaea. All right, the other thing that is unique about archaea cell membranes is, hold on. So our cell membranes and bacterial cell membranes are phospholipid bilayers. So you've got two layers of phospholipids, one on each side of the membrane. In archaea, they can actually have a monolayer. So instead of having a phosphate group and a fat tail, and then another fat tail and a phosphate group, there's just one fat molecule that runs the entire length, and it's got a phosphate group on either side. So one of the reasons archaea have membranes like this is they live in really nasty places, like boiling hot springs, or at the bottom of the ocean where there's, really nasty things. And if you have a single membrane like this, it's harder to rip your membrane apart than if you have a bilayer. Here the two layers could separate from each other if you heated it up or if there were toxic chemicals. All right, further inside the bacteria we have the cytoplasm, which is basically a jelly which is mostly water, but it has sugar, amino acids, proteins, and other things floating around in there. We kind of think of it as things floating around, but actually a lot of it is compartmentalized. There are cytoskeletal proteins like actin and myosin, and things actually kind of run along those tracks just like they do on highways for us. So the cell isn't just this bag of stuff floating around. It's actually fairly ordered and controlled. And then we also have our genome, the chromatin. We have ribosomes, fibers, the cytoskeleton. And then some bacteria can also have plasmids. So the genome of the bacteria is shown here in red, and you can have extra pieces of DNA known as plasmids. These are usually much smaller than the genome. So the genome of E. coli is about 4 million base pairs. A plasmid might be 10,000 base pairs. So what do these plasmids do? They frequently have things like antibiotic resistance or virulence proteins. And bacteria can share these from one to the other. So the Sex Pilus will share these plasmids from one bacteria to the next. The whole genome isn't shared through the Sex Pilus, just plasmids. All right, we like plasmids though because we can use them for genetic engineering. So we could take a plasmid from a bacteria and put something like the human insulin gene in it and stick it back into the bacteria, and now the bacteria will make insulin for us. And that's a whole lot cheaper than trying to purify insulin from cow pancreases and things like that. Just a lot easier. All right, ribosomes. So we mentioned earlier that bacterial ribosomes are a 70S ribosome and eukaryotic are 80S. These are found throughout the cytoplasm, and their function is protein production. So they take RNA and they use it as instructions to make proteins. All right, we can also have inclusion bodies. Inclusion bodies are basically densely packed structures. What's inside those can vary. So it could be a densely packed carbohydrate. It could be a densely packed protein. You can also have densely packed hematite. Who knows what hematite is? No one's ever had a hematite rock? Really?
Yeah.
What is it?
They make jewelry out of it. It's magnetic.
It's magnetic, yeah. So some bacteria actually use hematite as a compass. Literally, they use it as a compass to tell them where they're going. So we thought we were smart when we developed the compass like 5,000 years ago, but bacteria have been using compasses for like billions of years. So I don't expect you to necessarily memorize what could be in an inclusion body. Just know that it's a densely packed granule of something. It could be a sugar, it could be hematite, or something else. Sometimes they do have a membrane around them, but it's a single layer membrane, not a double layer. So if we go back to the cell membrane, instead of having a bilayer like this, just chop off the bottom and it would just be the top layer that's surrounding that instead of a double membrane. All right, then the cytoskeleton. So these are proteins that are long polymers. They can be arranged as a helix that kind of surrounds the cell. It contributes to the shape of the cell, and as I said, it kind of works as a highway to be able to control where things go inside the cell. All right, bacterial endospores. Some bacteria, not all, but some bacteria have the ability to make a spore. And the spore of a bacteria is different than the spore from a fungus. The spore from a fungus is like a seed from a plant. It's a reproductive mechanism. So the fungus makes a whole bunch of spores and spreads them everywhere in order to reproduce. In a bacteria, the spore is a survival mechanism. It is not a reproductive mechanism. So what happens is you have a vegetative cell, which is an active growing cell. If conditions get nasty, so let's say you run out of food, it gets too hot, a toxic chemical is present, instead of growing and dividing, the cell may decide instead to make a spore. So the sporangia is a temporary stage where the cell is making a spore, and in the end it makes an endospore, which is a resting condition. So once the endospore is made, it's not growing, it's not using energy, it just sits there. It's also very resistant to chemicals. You can put bleach on this for 10 minutes and it doesn't do anything to it. So this is also medically relevant. There are some spore-forming bacteria that are really a big pain. So C. diff in the hospital is a spore-forming bacteria. And the reason hospitals have such a problem with it is once it's there, it forms these spores, and then you can bleach a room, but you don't kill the spores. And so it just keeps coming back again and again and again. Okay, so watch this video on spore formation real quick.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell.
It's like the mothership.
Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolinic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
Okay, so the spore has now 2 cell membranes, as well as a cell wall, as well as like other coatings on it. And as it said, the dipicolinic acid causes all the water to leave the cell. Has anyone ever put something in a microwave that doesn't have any water in it?
Doesn't get hot.
It doesn't do anything, yeah. So the microwave actually heats things up because of the water molecules. And so if you microwave a spore, it does jack. You're wasting your time. So this is one reason that spores are so difficult to kill. So they've got all of these protective coatings. They have very little water. All right, so this is just picture form of what you saw in that video. So I do expect you to know the spore formation process. So you should know that you start off with a normal cell. You get duplication of the DNA. You make a 4 spore. You then have a double membrane. You put the cortex in there, which is basically a cell wall. And then some spores also have a coat or an exosporium around them. Any questions about spore formation? So the process takes 6 to 8 hours, depending on the species of bacteria. So this isn't like an instantaneous process. So if you're a spore-forming bacteria and conditions turn nasty, 6 to 8 hours is a long time. So if you dump bleach on this bacteria, bleach is going to kill them faster than 6 to 8 hours. Then how are they protected? Well, what happens is some of the bacteria are always making spores. So even if conditions are good, there's a few bacteria that are always making these spores. And so even if most of the population gets wiped out by bleach or something, a few of them still survive because they were always in the process of making some spores. All right, how does the spore wake back up though? Exposure to water and germination agents, so this would be sugar molecules or something. Now the bacteria, the spore knows that conditions are good, there's water again, there's food, and so the spore wakes back up. It basically has to cut itself free though, so it has all of these protective layers, and so it makes some enzymes that chop all of those protective layers away. And then it basically returns to a normal cell. It starts growing again. The germination process takes about an hour and a half. Right, so I mentioned C. diff as a medically relevant bacteria. So it causes something known as pseudomembranous colitis. So basically you get a really nasty intestinal infection from it. Anthrax. You've heard of anthrax spores. Tetanus. So you get your tetanus vaccine so that the spores from tetanus don't get in you and cause lockjaw. Clostridium perfringens causes gas gangrene, and botulism is caused by Clostridium botulinum. So all of these medically important bacteria are spore formers. Now some of them, like tetanus and botulism, they're just environmental bacteria. They usually grow in the soil, and they don't usually cause problems for humans unless you get a cut. And then once the spores get into your cut, they grow and they produce really nasty toxins. So it's actually the toxin from tetanus and botulism that cause a problem for you. All right, so I mentioned they're very resistant to things like bleach. You basically have to soak a spore in bleach for like an hour before it's killed. So no one does that. You're not just going to flood a hospital room with bleach for an hour waiting for all of these spores to get killed. They can also contaminate cultures and media. We try and protect against spores getting into wounds by using good sterile technique when we're doing surgeries and things. Botulism is a big problem for food canning. So when canned foods first came out, kind of mid to late 1800s, they would have problems with botulism because those spores would just be in the soil, and so they would get on your tomatoes or whatever that you were canning. And then if you didn't can it properly, the spores would germinate, produce the toxin, and then when you ate the food, you would get botulism. And basically all the muscles in your body relax, and you can't breathe anymore because your diaphragm stops working. So not a very friendly disease. All right. Another quick web clicker question. So what's the difference between a bacterial ribosome and a eukaryotic ribosome? All right, so the bacterial ribosome is smaller and less dense. So 70S means it's smaller and less dense. All right, let's finish up this lecture quickly. The archaea, archaea are actually more similar to eukaryotic cells than they are to bacteria. So even though their cell membranes are different, a lot of other things about archaea are actually more like us. The ribosomal RNA sequence is more like eukaryotic cells. Their protein synthesis pathways are more like us. They are different, though. We talked about the cell membranes being different. And they also have a unique method of DNA compaction. The archaea, some scientists think that that's probably one of the first types of cells that appeared on Earth about 4 billion years ago. And the reason for that is we find them in such nasty conditions. They can grow in acid. They can grow in hot places. They can grow in high salt concentrations. All right, so this table just kind of compares the bacteria, the archaea, and the eukaryotic cells. So look through this table, make sure that you know what the differences are between the cell membranes of the ribosomes and other things. Right. Diagnostic scheme. So if you are in a medical profession and you need to make some type of diagnosis on an organism that's causing an infection, usually one of the first things you do is determine, is it gram-positive, is it gram-negative, or is it weird and it has no cell wall, or it's like tuberculosis and it's an acid-fast bacteria. You then look at the cell shape. Is it a coccus? Is it a bacillus? You look at the clustering. Is it a streptococcus or is it a staphylococcus? Meaning, is it a long string of cocci or is it just clusters of cocci? Oxygen usage. Is it aerobic or anaerobic or facultative? Meaning it can use oxygen or live without oxygen. So we're going to skip through these slides real quick. I will do that. All right, true or false? Archaea do not cause human disease. Is there any disease caused by an archaea? Actually, there's not. I've never heard of a disease caused by an archaea. Diseases are caused by fungi, bacteria, parasites, but there's actually, as far as we know, no disease that's caused by an archaea. All right. So let's keep going with lecture 4. All right, we got 5 more minutes. So the eukaryotic cell. What you should know. Again, you really want to focus on the similarities and differences between eukaryotic cells and prokaryotic cells. You should know the cell membrane, cell wall, ribosomes, whether they're similar or not between bacteria, archaea, and eukaryotes. Eukaryotes have organelles. They have things like mitochondria and chloroplasts. You need to be able to recognize what those look like and what they do in a cell. All right, eukaryotic cells can be single-cell organisms like an amoeba, or they can be multicellular organisms like you and me. What we think happened is somewhere along in the evolutionary history, we had a biofilm. So it was this kind of sticky mass of bacteria, and eukaryotic cells in this case. And some of the eukaryotic cells started doing different things than other eukaryotic cells. And eventually that became so important that you formed tissues. You formed a liver. You formed a brain. You formed muscle. And that's how you ended up with a complicated organism. All right. Protozoa. These are always unicellular. Fungi and algae can be unicellular or multicellular. And then helminths, so these are things like tapeworms, those are always multicellular. So these are your kind of relevant organisms for microbiology. So the general characteristics of a eukaryotic cell, there's always a nucleus. There are membrane-bound organelles like a mitochondria or a chloroplast. You have a cell membrane on all eukaryotic cells, but only some cells have a cell wall. Most eukaryotic cells are also bigger, so this is the average size of a bacteria compared to the average size of a eukaryotic cell. All right, the flagella. So bacteria have flagella, and some eukaryotic cells have flagella, but they don't function the same way. So remember, in the bacteria, the flagella works like a motor. It looks like a prop on a boat or something. It spins around. In eukaryotic cells, there's actually a cell membrane around the flagella, and you have microtubules inside. We call the microtubules in a 9+2 arrangement. There's 9 around the outside and 2 on the inside. And the eukaryotic flagella is more like a whip. So it moves through the water by moving back and forth instead of spinning around like the bacterial flagella. Cilia. So you have cilia in your lungs and in your intestines, and it helps move mucus. Microscopic organisms use cilia as a way to feed or move. So some groups of protozoa have cilia. The glycocalyx. It looks like the hair on top of this cell is part of the extracellular matrix. It's usually composed of polysaccharides. It can be fibers, slime layers, or capsules, just like in bacteria. All right, we'll go ahead and stop there, and we'll pick up on cell walls next week. Have a good Labor Day weekend. Don't forget to do your reading assignment, though.
Are you going to give us a study guide for
for the exam. What I'll do is I'll take all of the clicker questions as well as additional questions, and I'll make like a pre-test on Blackboard, and you'll be able to go through that pre-test.
So are the questions going to be like questions, like structured like that?
Some of them will be. So it'll all be multiple choice or true-false questions. Yeah.
We were talking about how a cell wall of bacteria can be destroyed by penicillin.
Yes.
What if someone's allergic?
Then you can't use penicillin.
I'm allergic to everything in the penicillin family.
Right.
So.
There are other antibiotics that attack the cell wall. I'm trying to think of one. It's not coming to mind. Anyway, but there are other types of antibiotics that attack the cell wall.
Now I know, like my biomedical sciences class, the tests are all on our clicker,
Right.
like just the actual clicker.
Oh, yeah,
Clicker
no, that's not.
questions.
Yeah. So this, yeah, you'll have a Scantron sheet, and you'll fill in the Scantron sheet. So,
That's how we did it for my chemistry class.
yeah. But
Well,
that's,
at my junior college.
yeah, that's how I do it. It'll just be a Scantron sheet.
I sent you an email, but it was about the Connect,
Okay.
and I got everything correct on it except for when it said when I was doing the streak plate,
Oh, yeah.
and
I
it was
remember
just
that.
like going around. It was like not letting me do anything. It let me do one streak.
Some of those are weird, yeah.
So I don't know like how that affects my grade or what. I know I got like it was.
Were
so
you
much
able to
percent
complete
off.
the whole thing?
No, it didn't say the percentage. I got a lower percentage because they wouldn't let me do the quadrants.
Right.
And I mean, I went like over that, and I tried to do it 4 times.
But were you able to complete all the sections?
I did all the sections.
Okay, that's fine. So I'm not grading those. Like, if you do it, you get credit.
Oh,
is the way-
okay. Okay, cool.
Yeah.
All right,
There's a
awesome.
little arrow around mine.
Yeah, seeing
After I
it has.
scratched it, it would put an arrow to let me rotate it.
Oh.
It was hard to see though.
Well, I mean, those are helpful. I really like them.
Yeah,
For
no.
lab, that's really good.
The thing is, where you guys only have lab once a week, like, I think it really helps you just be
Yeah.
like, okay, this is what I'm going to be doing.
I wouldn't
Okay, so it doesn't matter. Great.
so
Okay,
lost
great.
without that
Okay,
thing.
cool. Thank
But
you.
don't just, like, blow them off because they're...
Oh, I won't. I'll do them. It helped a lot.
Okay.
This glycocalyx,
Yes.
that's inside of this S layer. Is that part of that S layer? 'Cause I'm just
The
looking
S
for
layer
it
is
on this.
different, yeah. I don't know that it shows it on there.
Would it be like in between these two, or I would just try to figure
Usually
out.
they have one or the other. They'll either have an S layer or they'll have a glycocalyx. Yeah.
Did I get this? The